In our previous video, we introduced enzymes, and we said that enzymes are almost always proteins. So I want to show you a real protein that is an enzyme. In fact, I've shown you this one before. It's a beautiful protein. It's one we work on in the lab, and it's an enzyme that breaks down penicillin-type drugs, and it makes it very dangerous. You can see beautiful yellow beta sheet strands in the middle. Nice red alpha helices and green loops. I also want to show you something unique. Even though this is a relatively globular water-soluble protein, there's a gouge or a groove right here that goes deeply into the center of the enzyme. In order to understand how enzymes work, we have to start with this groove, and we'll do so on the next page. That's called the active site, and the active site is what this whole video is about. The active site is a groove on the surface of the protein that is an enzyme. And in order to understand this, what we have to say is that the active site is going to, or must, complement the substrate. Now, what do I mean by complement? Well, first of all, let's note that that is a complement with an E, not with an I. This is not the case of the enzyme seeing the substrate and saying, hey, you look great. Not that kind of complement. This is complement with an E, and that is going to be fits. Fits like a hand in a glove. Now, to understand how an enzyme and a substrate fit each other or complement each other, we can say that it's all done by chemical means. In fact, it's done by the same types of chemical means that we've talked about before. If you have a positive charge on the substrate or the reactant that you want to break down, it's going to be complemented in the enzyme active site by a negative charge. A negative charge on the substrate will be complemented by a positive charge in the active site. Nonpolar groups, see if you can guess what that is. Nonpolar groups on the substrate will be complemented by nonpolar groups on the enzyme. Remember, nonpolar goes together, and the hydrophobic is going to draw them to cluster with each other. Hydrogen bond donors on the substrate will be complemented by acceptors, and acceptors will be complemented by donors. So we practiced all that stuff for a reason, and here's one of the reasons that we want to understand them. There's more to complementarity, though. In fact, the most important type of complementarity is simply shape, and I'll include another part of that, chirality. Let's think about what I mean by shape. Let's consider how a hand fits in a glove. We know that that's not a chemical thing, but certainly if we have a glove that has 4 fingers and a thumb, and your hand has 4 fingers and a thumb, they're going to fit in there beautifully. But let's not think about a regular glove. Let's think about something like a hockey glove. A hockey glove has 4 fingers and a thumb, but if you try to put your right hand in a left-handed hockey glove, it's not going to work. But if you try to match its chirality, that is, you put a right-handed hand into a right-handed glove, then you're going to get a good match. So the shape and complement and chirality must be correct. They must match. So we're going to practice this out a little bit, and we're going to do some homework on this, but we're also going to just show you how this might work by looking at an example down here at the bottom of the page. Here is an example of an active site, and what you got to understand is the active site is this large enzyme here, or it's in the large enzyme. This is the protein down here. And, that's all the protein structure. Then we have this beautiful groove here, and the substrate fits in it. Now, this is another enzyme that we talked about before. This is called trypsin, and I want to introduce you this very interesting topic. Trypsin is another enzyme that is in your intestine, and you may remember that it breaks peptide bonds, like chymotrypsin did, but it does so with a different type of specificity. Remember, trypsin cuts peptide bonds after long, narrow, positively charged residues like lysine and arginine. So let's practice this. I want to give you a real life example of something that happens in some people's intestine. We have trypsin, and this is actually not a protein that it needs to cut. It's actually a drug, and the drug is called Vyvanse. It's a common drug. Some of you may even take it. It's used to treat ADHD. And I'll tell you how it works in a minute, but what I want to do is tell you that it's activated in your intestine to an active form by the enzyme trypsin. And what it does is it breaks the peptide bond between these two species and releases the drug. So what I want you to do, though, is to practice here a little bit. What I'd like you to do is at the point I put these red dots, I'd like you to draw the side chain of an appropriate amino acid, 3 in all, and I want to see if you can complement the structure, the chemical properties of Vyvanse. So go ahead and pause and see what you come up with. Okay, I really hope you gave this a try. Let's start with something over here on the right. What might we use to complement this phenyl group over here? It looks like a phenylalanine. It's not quite a phenylalanine, but what could we use over there? Well, let's see. We'll use a red pen here for this. We could use something like a leucine. A leucine would be beautifully complementing that phenyl group because it has nonpolar interactions. Okay, very good. What about down here on this carbonyl down here that's part of the peptide bond? Maybe we want to complement that. Well, I might suggest that we use something like a serine. And what we end up with is a beautiful donor-acceptor relationship here where the serine is a donor and the Vyvanse carbonyl is an acceptor. That's a good complement. What about up here on the NH3? Well, you could probably guess that it might make sense for us to put some kind of a carboxylate negative charge like a aspartate. And that, of course, will be ionic interaction. Down here we said hydrogen bond. I forgot to write in that this was a serine. Okay, so this is how it works, and we'll definitely be practicing and testing you on this kind of thing, that complementarity between the substrate and the active site is critical. By the way, let me just finish off by telling you about this drug Vyvanse. How does it work? Well, Vyvanse is actually amphetamine, which is a powerful stimulant. It can be abused. It's what's in Adderall. And Vyvanse has got rather the same thing from Adderall right here in the green, but cleverly the drug company attached it by a peptide bond to a lysine. The entire drug, which is shown, the whole thing, that's Vyvanse, you take that orally, it goes into your intestine, but it's not active because it's got this big lysine attached to it. But then the trypsin in your digestive tract slowly, over time, breaks the peptide bond, because that's what it does, and it releases the green part, which is the amphetamine. That it's a slow release, non-abusable version of something very similar to Adderall. Okay, in any case, that's an example of how an enzyme can interact and complement a substrate.